What gets into our body and changes our health, it's literally thousands of chemicals. If you want to explain complex diseases, you need to look at all the things that contribute to it.
There is a study of the fascinating interplay between the cumulative effect of all the exposures on a body and our genes. It's called exposomics, and that is the topic on Tomorrow's Cure, a podcast from Mayo Clinic that brings the future of medicine to the present. I'm Kathy Werzer. Thanks for being with us. We are talking to two leading explorers of the exposome today. We're so excited about this. Dr. Gary Miller is an international leader on the exposome. He founded the first exposome research center in the country. Dr. Konstantinos Lazaridis is a gastroenterologist who is the Carlson and Nelson Endowed Executive Director for Mayo Clinic Center for Individualized Medicine. So I'm wondering, what specific exposures make up the exposome?
So the exposome is designed to capture as many of the exposures as possible. And it's not just the ones we would think that would be negative, it's the positive ones as well. So when you eat your breakfast, there's a lot of nutrients and vitamins there. That's good. But there might be pesticide residues or plastic residues on it. You might have air pollution or allergens in the air that you're breathing. And so what we're looking at is all the things that people get exposed to. And I like to think about it of the compilation of all the physical, biological, chemical, and psychosocial influences on our health. I think that's an area that it's been harder to study because it's not something you can just measure with a instrument of some sort. But I think that's a key part of it because we know that these complex social factors have a big influence on our other exposures and how our body responds to those.
Well, we've talked a lot in the news about generational trauma. Is that folded into this in a sense?
Absolutely. I think this is where, again, like past traumas change our biology. And those are things that can stick with us for a long time. And that's part of our exposome.
Dr. Lazaridis, if I recall correctly, when you started at Mayo Clinic, you were studying the genetics of liver disease. What were some of the things that you found that were the basis of your research?
Yes. So at the time that I started, I was intrigued by the Human Genome Project. The Human Genome Project provided a framework to study genetics. And as you know, in science, we're as good as the tools we have to apply. And so at that time, 25 years ago, this was something possible to be measured. And we believed, and we still believe, that genetics are significant contributors to liver disease. But at the same time, what captured my attention as I was studying families with liver disease, we realized that those families, these family members, they don't share just the genetics. They share a household, a diet, activities, the water they drink. And I thought, okay, we have their DNA material. We can measure what is shared genetically, but how can I measure the environment? So naively, I started thinking about epidemiological studies. What do you drink? What do you eat? But we never measured anything. And we found very little on the questionnaires, because also there is the recall bias when you ask these questions. And at that point, I was exploring new opportunities, and I went to the web and I asked about environmental exposures and liver disease. There was nothing there. And then in this context, I found what Dr. Miller has developed. And I said to myself, I will call this guy and we'll do something, because now he can measure things on the bloodstream. That's how it started. And that was almost 7-8 years ago.
You're kidding. You picked up the phone and you called him.
Yeah. And he invited me to Emory. And the week after, I went there, talked about it, got excited. We wrote grants and we kept collaborating. And what we found that the patients with this chronic liver disease, they have a significant exposure to a fungicide that we found it because we measure a lot of exposures in the bloodstream. And as I said earlier, on the questionnaire, there was nothing there because we don't.
The patients wouldn't know that.
We don't know that, yeah.
What did you think, Dr. Miller, when Dr. Lazaridis from Mayo called and said, hey, I'm doing this work?
I typically don't answer my office phone, so it was somewhat fortuitous. But what I was most struck by initially in that call was that he asked the questions about the expose and wanted to learn more. And like he said, he was in Atlanta the next week. And so I was like, this guy means business. He wants to figure this out. And our technology just got to this point where we were ready to really dig into this analysis, where like you can go to your doctor and get a blood test for cholesterol and your clinical chemistry. But with the new technology, we can measure thousands and thousands of molecules in blood. We weren't looking for the fungicides in that study. We were just looking for molecules. We were able to identify them later what they were. But the idea is to get that comprehensive analysis the way that geneticists do it for genomics.
Now, we know the risk of developing chronic diseases are attributed to both genetic and environmental factors, right? I'm curious, which plays a more important role in disease development? What do we know about that?
So a shorthand rule that we often use is that for chronic diseases, we can only explain about 30% of the variation purely by genetics. And so that means the non-genetic components and potentially gene-environment interactions make up the rest of it.
Wow. You hear this often. I think some people have a misconception that our genes are set in stone, but research shows that early experiences can determine how genes are turned on and off. How does the exposome contribute to this?
Yeah, there's a great example is that when we think about our past exposures, you may wonder, like, how can you measure something that happened 20 years ago? But there's actually really good data with epigenetics where people that smoked 30 years ago and quit, you can still see these epigenetic marks on their genes 30 years later. And so the idea when you think about how a past childhood exposure might kind of imprint you and keep going is that the epigenetics is probably a key role in that.
We can measure the biological age of an individual by assessing the markers on top of the genome of an individual. And it's not uncommon these days to find out that the biological age of an individual, it may be years above or under the chronological age. And this also gives us an opportunity to think about chronic diseases. Can we reverse this process? If we reverse it, is this going to help the disease? We can measure this today, and it correlates with many chronic diseases, whether it is cardiac disease, whether it is digestive diseases and the likes, which is something for us not only to know, but to understand the biology behind that.
Dr. Miller, this sounds like the exposome, what we're talking about here, sounds like this is a paradigm change in medicine. Am I right about that?
Yeah, I think that for many years we've studied one marker at a time. Like, you think about lead exposure over the past 50 years. We knew that lead was bad. We removed it from gasoline and things and saw improvements in IQ at a population level. The challenge now is that we need to recognize that we're exposed to hundreds or thousands of things. And the idea is that our systems hadn't been built to study that complexity, but that's what we're trying to do with exposomics.
When you're talking about so many exposures over a lifetime, how do you even begin? It's like a needle in a haystack, isn't it, in a sense? There's just so much data.
The starting point is hard because unless I have a sample from you when you were a child, I don't have that. But I do have a sample of blood from you today, and you are who you are today. And so that was the starting point, was just you work with the samples you have. Our measurement technologies have improved so much in the last 50 years that, again, we can measure thousands of things at a time now.
There's exposure, if I'm remembering this, to iron and copper in liver disease, right? This is all, again, part and parcel to the exposome, correct?
So remember, there are elements that nature created for us to have biological function. And some of those, just mentioned copper and iron. We know also that there are some people who genetically absorb more iron than others. And there are some individuals who cannot secrete copper compared to others. And so then, whether absorb more iron or whether you secrete less copper, this ends in your liver. And accumulation of those natural elements creates disease. We call this hereditary hemochromatosis for the iron. We call this Wilson's disease for the copper. So those are two innocuous chemicals that, if accumulated over a long period of time, they lead to disease, they lead to cancer, they lead to liver failure. So conceptually, in my mind, I say, well, if nature-made elements can cause disease with all good intentions, imagine what can happen if humans, we produce all these chemicals for which we don't know what they cause when we develop them. And at the same time, as we have now ways to treat people who have accumulation of iron or accumulation of copper, if we discover it early, why not we can do the same thing for pesticides, for chemical exposures? I think, Kathy, one day in the next 5 to 10 years, we will develop ways to screen individuals based on their exposures. I would like this to be, that's my dream for medical practice, to be able to screen exposures at large and then creating plans of care where we say, you are accumulating this molecule. We have to find some ways to reduce this from your system, because if this continues, we'll have a problem.
Yeah, people have discussed the idea of a human exposome project, and the challenge is with the Human Genome Project, you had a finite number of base pairs you had to sequence. With our exposures and even across a lifespan, it's much more complicated. But what we can do is we can look at what are all the chemicals and small molecules that are there that humans are exposed to. The idea, what is like the normal reference range for everybody, and then see where people plot along that curve and if there's associations with various diseases. So to me, it's going to be kind of an ongoing process. It'll get more and more detailed, but there won't be a clear finish line to it. It'll be something where, like, for example, 10 years from now, our exposures will be different than they are now.
I asked Dr. Lazaridis about how he began, and I want to take you back just a little bit in your professional career. You started the Center for Innovative Exposomics at Columbia, and part of your lab's focus is on different set of illnesses, neurodegenerative diseases like Parkinson's and Alzheimer's. What shift happened in your own thinking way back when that got you thinking, there's something here, especially when it comes to Parkinson's? What am I looking at?
Yeah, so I started my work on Parkinson's about 30 years ago, and one really thing that stood out about the disease was that people that lived closer to farms and in rural communities had a higher incidence of Parkinson's, very much the opposite of cancers and things. And what people had hypothesized was that pesticide exposure was connected to it. And my background's in toxicology, and my first reaction was that there's like hundreds of different pesticides. Like, what does it mean to say pesticides? Because you have things to kill rats and bees and mosquitoes and all these, and even plants. And so what I saw, what the field was doing is that they were studying one chemical at a time, and if they saw an association in a laboratory animal, they report it. And I'm like, that's not a comprehensive and systematic way to study a disease. And so while I worked on that for a while, when I was introduced to the concept of the exposome, I was like, that's what we need to study a disease like that.
We've been focusing on pesticides, chemical exposures in our conversation to this point. But I'm wondering, and when you talk about exposures over a lifetime, are we also talking about socioeconomic issues that one might have in their life, stressors? How might all that fit in? And I don't understand how you would even measure those stressors.
Yeah, I like to consider the exposomics covering all of the non-genetic factors. And so socioeconomic factors, lifestyle factors, your access to parks and things should all be under this.
Racism?
Structural racism. It's a good example is that we know that structural racism can lead to adverse health outcomes. But if there's an adverse health outcome, there had to be a biological underpinning to that. And the idea with exposomics is we want to look at those things. So, for example, if you think about, people that live in a poor community, why is it that their health outcomes aren't as good? Is it because of lack of access to care? Is it because they're closer to the highway and have more air pollution exposures? But the idea is that these things have to get under the skin. They have to get inside the body to change biology. And that's what we're trying to do is by measuring blood samples in the context of these sort of social structures, we can make those connections. And so for a socioeconomic piece, maybe we don't find the molecule causing it, but we find the molecules that are mediating it. We see the increased stress pathways.
I think Dr. Miller made a very good point that I like to reiterate is it's the connection of the exposure to the biology. There are millions of exposures. What are probably the most important ones are those who are connecting to the biology. Because if there is an exposure and there is no connection to biology, but I want to know about those exposures that affect my immune system, they affect the way my organs are aging, they're affecting my mechanisms to prevent or promote cancer. I want to know those. And if I'm aware of those things, I can do something about it. And so learning about your exposures, it can affect your way of thinking about preventing disease. You may start doing things which you don't do because now you know that you can do something about it. And this can be simply changing your diet, changing how you prepare your food. Something that is very hot these days is micro and nanoplastics.
Yes.
Correct? At large, we don't know how those exposures affect our biology, but I'm more cognizant now when people are reporting that those microplastics may be relevant to cardiovascular disease and the inflammatory reactions. And I say, I'm not going to use a plastic bag when I go to the grocery store. I can use a paper. I'm going to drink water from a metallic container or from a glass container. I care about all of us exposing so much to elements that we don't know what causes, disease, particularly for the young generation. Because those things, as Dr. Miller taught me over the years, the dramatic exposure that happens once in a lifetime is those small events that happen every day over 5 years, 10 years that created a problem. How can we reverse that? Several decades ago, everybody was smoking.
Yes, right.
Correct?
Right.
We can change public behavior if we understand the connection. And this can save lives. We see a lot of people, compared to the past, developing colon cancer and lung cancer at a younger age. Genes cannot explain that. Our genes are not changing this fast. I don't think it's our genes. I don't know what it is, but we have to pay the attention to identify those other non-genetic elements to that.
How might our exposure to a warming climate, more heat waves, how might that affect us? Do we have any idea, Dr. Miller?
So I like to think of climate change and the sort of variations we have in overall temperature patterns as being a force multiplier to all of our exposures. For example, if you think, if you've ever tried to stir sugar into a glass of iced tea versus hot tea, the warmer things are, the more chemical interactions occur. And we live in a soup of chemicals. And so when the temperatures get hotter, you're creating more opportunities for those interactions to occur, and you're exacerbating a lot of underlying problems that are already there. So the climate change is really just making many things worse.
We know about lung cancer and being exposed to cigarette smoke and that kind of thing. I'm wondering about just generally speaking, exposomics and cancer. Are there big studies around that at all?
Yeah, so recently a colleague of mine asked me to participate in a study that was trying to figure out why African Americans have a higher incidence of cancer. And they wanted to really go deep with this. And so what the study is designed to do is to look at people from 5 countries in Africa and people of African descent in the US and the UK. They're going to do whole genome sequencing but they're also going to collect information on social determinants of health and exposomics. And the idea is when you can combine the social, environmental, and genetics together, it'll really help kind of figure out why we have these disparities in outcomes.
And that's not been done before.
Right. I mean, I think this is something we know from the genetic side is that our databases are underrepresented from underrepresented populations. And what if you're primarily a white population, the findings may not be generalizable to other populations. And so I think now we're seeing with exposomics, we need to have that same level of coverage of different parts of the world and not just in the European and American. We need to be in South America and Africa and India and China to really figure out what the real whole range of exposures that we have across the world.
I want to go back to the Human Genome Project for just a moment. We mentioned that briefly. What do you both think could be learned from that experience when it comes to exposomics? Not only what you could learn from that, but also some of the pitfalls that may have occurred during all that work that went on for the Human Genome Project. What do you think, Dr. Miller?
There's actually people have written papers about lessons learned from the Human Genome Project, and I've read them carefully. One of the best examples I saw was the idea of how to break down the problem. The idea is that we need to systematically study the exposome, and what is the best way to do that? I'd love to have a study where I had annual blood samples from a population, but if I don't have that yet, I start with the existing samples. Maybe we have them every 5 or 10 years. But I think as we just need to show the progress and kind of getting deeper and deeper as we go forward. But that was a key part of the Human Genome Project was how they staged it out and had very clear milestones.
Dr. Lazaridis?
I always realize that not every exposure, for the most part, will bring the same risk to every individual. But there will be a particular host, a particular individual who are more sensitive to a particular exposure than another. And to identify this interaction, we're going to need to have the genome sequencing available. And so imagine what this will bring to us investigators and to the public down the road. We can say this particular exposure is not going to be harmful to you because you're not going to react to it compared to another individual. So that will be part in my dream of the approach we'll take. I can see the time where you come to see a physician and they will screen your genome and your exposome. And what happens after that for your preventive care will depend on those two elements. Of course, you may have a question or a condition for which you come to have an evaluation. But I think the major contribution of medicine will be, if this is my genome and that's my exposome history, what can we tell you about the future?
One of the new projects we have together is to look at people's genetic background and their exposome to see how it affects the drugs that they take. So, many people take 2, 3, 4, or 5 different drugs, and we know many things in the environment can affect how they're metabolized. You probably know that grapefruit juice can interfere with many drugs. There's many other things besides grapefruit juice that interfere with that. And the idea, if we can measure these in a systematic way in large populations, we can identify other types of exposures that may decrease the ability of the drugs to do their job.
So as you work on this, I'm wondering about what kind of standardization, cross-disciplinary collaboration might have to happen in order to move this forward. What do you think, Dr. Miller?
You mentioned the Human Genome Project before, and I think there's a lot of very good lessons there, is that until the different laboratories could use the same system and get reliable data at different sites, it was very hard to combine it. We're working on the same things with exposomics, is that we have to know that a lab in Rochester, Minnesota and New York City can get the same results. And so we've been working very hard on that harmonization of the research platforms.
What about, by the way, getting researchers fired up about this? I mean, especially young individuals coming into the field.
I think when you talk about exposures and environment, it excites young people because they know about climate change. This is the This is a place where they're going to live and grow for the next, 50, 60 years or more. I think young people, young investigators, they're very excited about the topic at large, because they like to create also, be part of the solutions, create opportunities to learn about exposures.
Yeah, I'd say if you go back like 10 or 15 years ago, people were still thinking that as the genomic techniques got better and better, it would close the gap. And I think we've reached a point now where the genetics has explained a lot, but there's this gap there. And so it's like, what's the rest of it? And that's where exposomics fits. And I think that's why many younger investigators, they see it as, if you want to explain complex diseases, you need to look at all the things that contribute to it.
As the exposome becomes more integrated into research, I wonder how you both think that the clinical practice may change, perhaps, even 20, 30 years down the line.
To me, like, I imagine a clinician seeing a patient who they've tried on a drug and they didn't respond well to it, and that they can draw a blood sample, send it to the lab, and then a couple hours later they can say, here's why you didn't respond well to it. We should put you on this different treatment.
I like to observe nature because it tells me many times the secrets of life and what makes a difference. And if you think about the blue zones, there are places on Earth where people live longer.
Yes.
I don't think it's about their genes. I think it's about the environment they live and the activities they have and the socio-economic interaction they have in those places. And so we're going to learn from nature again if we go to study exposures and populations. And I think part of this research we may lead to us to understand what protects from disease. And so that's a positive, point I like to make. Let's find out those exposures that gives us wellness, gives us health. They're around us, but we have to dissect them. We have to study them. And as I'm thinking about the medicine of the 2030 and beyond, it's about creating this knowledge about your genome blueprint, which you cannot change for the most part at the present time, and your exposures, which I think you can do something about it, either to promote the ones that are good and you are exposed to, or to reduce the ones who are harmful.
I'm wondering, with what you know, have you made any personal changes in your lifestyles at all when it comes to what you eat and drink, what you expose yourself to?
I spend more time to think about my diet, and I'm shifted to more organic diet, more natural processes for cooking food, and less exposure to plastic. I know it sounds very small, but if we all do this, it will have an effect.
Dr. Miller?
To me, I think about the packaging of things. The more processed our foods are, the worse they are for us. And so your own habits of consumption, like mentioning like the idea of water, like I only drink out of glass. I don't buy plastic water bottles anymore. Another thing that I think came up during COVID was we knew how well a lot of these HEPA filtration systems worked. And we were concerned then about a virus, but it also removes pollen and small particles and various volatiles. And so I routinely recommend to people that they should have HEPA filters, especially in their bedrooms where you're sleeping at night, because it reduces your exposures to many things that aggravate us.
Getting back to stories in the news, forever chemicals, PFAS. I mean, there are a number of different things that are going on in the environment. And with the study of the exposome, I'm wondering, can this be fodder, perhaps, for lawmakers to make long-term public policy changes, perhaps?
Yeah, one of the challenges we have now in the regulatory space is that we can only regulate very specific chemicals. And so there's been recent moves on PFAS, where there's a limited number of chemicals, just a handful. But there are literally thousands of variants of fluorinated compounds in the environment. And right now we're playing regulatory whack-a-mole by just picking out a few of them at a time. Now, the organizations that make the products, if you have a regulatory change there, it could affect hundreds of those exposures. But what gets into our body and changes our health, it's literally thousands of chemicals.
What do you want the general public to be thinking about when it comes to the exposome?
A big part of our exposures is diet. We should all use less processed food. Those are common sense things that our mothers and fathers tell us, but that's the reality. There are some exposures that are useful, that are helping us by preventing disease, and we like to discover those too. For example, what we eat, what we drink, it can protect us from disease too.
Dr. Miller, what do you want folks to be thinking about?
I think it's important to think about your surroundings holistically. So when you go to your day-to-day life, you think about how you're consuming food. If you have to spray a hornet's nest, you put gloves on before you handle a pesticide in your garage. You protect yourself from these potential exposures. We know that our genes and environment determine our health. And we've done a great job of studying the genes, and we have to do a better job of studying the environment. And we need to do it for all of our diseases and for health, for looking for optimizing health. My hope is that exposomics puts the environment on that equal footing to the genes.
I have enjoyed the conversation. I think it's absolutely fascinating. Thank you for your work on the exposome, both of you.
You're welcome.
It's been a pleasure.
Thank you.
We've been talking to Dr. Gary Miller and Dr. Konstantinos Lazaridis. Subscribe to Tomorrow's Cure on Spotify, iHeart, and Apple Podcasts, or wherever you get your podcasts for future conversations.